Hello, everyone, and welcome to the first of our Hot Topics in Imaging series, the first of this year anyway, of 2021. We're really delighted to welcome Charles Manning, who is somewhat new to MD Anderson. You should have in your email kind of a little bit of a more detailed bio, but I was so impressed reading through this and completely convinced that he's going to be an asset to the medical center and great opportunities for many of you on this call to maybe reach out and pursue collaborations in the future. Just briefly, Dr. Manning's a chemist. He's got a background in radiochemistry, medicinal chemistry, and imaging science. He comes to us from Vanderbilt. He was there since 2006, where he worked on chemical and molecular probes for cancer imaging and therapy. He served as the Ingram Cancer Center at the Ingram Cancer Center as the director of cancer imaging from 2015, and he was actually the founding director of the Vanderbilt Center for Molecular Probes and Molecular Imaging Research. And then this past year, his lab relocated to MD Anderson, and there he is a professor in the Department of Cancer Systems Imaging and serves as the scientific director of the Center for Advanced Biomedical Imaging in the Division of Diagnostic Imaging. He is a CPRIT scholar. He's had continuous NCI funding. And currently they're focused on new small molecule therapies targeting glutamine metabolism in oncology and other diseases, and developing new radiochemistry technologies, and I'm sure many other things, some of which we'll get to hear about today. But again, Dr. Manning, we're delighted to have you. And with that, I'll just turn it over to you.
Well, I just thank you so much, Suzanne and Dawn, for helping arrange, and all Jim and others who have gotten me linked up with this group. And I told Jim Bankson that I was so excited to be a part of the Gulf Coast and present to the group. And today, I think I'll just give kind of an overview of some of the programs and efforts that we've been engaged in as we've transitioned from Vanderbilt to MD Anderson. And of course, we're launching a number of new initiatives, and I'll warn you that some of this is work that it goes back a few weeks or months because of the obviously the pandemic has slowed us down to a certain extent. We're just so excited though to be here and to meet so many new people, and I would certainly welcome, please email me or reach out in any way if you're interested in any of this work, and I'll be happy to follow up at any time. Let's see if I can. Okay, so just a few technical disclosures. And so our work is sort of motivated by and large by sort of identifying a path to sort of precision health or precision cancer medicine. I am very excited to be at MD Anderson, where we focus almost exclusively on cancer. This has been our main focus in our lab for some time. But identifying that path is so critical in maintaining our route on that path. And what I hope to convince you over the next 45 minutes or so is that really a great path to precision health and medicine goes through PET tracers. And I'll give you some examples of where we've utilized or developed PET tracers to drive or realize some diverse areas of research, which includes both diagnostic purposes and therapy. And again, we're both motivated and frustrated in this line of research that making images and seeing things are just not enough. It's great that we can identify tissues of interest, but we'd like to really understand through the use of molecular or potentially other techniques, what are some of the underlying molecular or otherwise features that tell us about the disease or the tissue of interest? What are the features that might help us to understand, would this metastasis in the thorax be likely to respond to a certain type of therapy? And really, this is where we'd like to go. As we all know in this group, molecular imaging and PET overall are very well suited for precision medicine, precision health, to be able to address these questions with quantitative imaging gives us a unique opportunity to impact healthcare. So as was mentioned earlier, we have focused a great deal recently on glutamine and glutamine metabolism, and it's just a fascinating area of research in my opinion. And of course, we know a number of sort of characteristics of glutamine metabolism in oncology, but I'll just summarize a few here. Certainly a hallmark of many cancer cells. Glutamine is quite promiscuous in how it can be utilized by cancer cells. Some of the notable features are biosynthesis, certainly, but we're learning that ROS is also critically important, and actually targeting the reactive oxygen species pathway is becoming very interesting. A number of individuals and companies have gone after glutamine metabolism as a therapeutic approach, and we've played in this area as well. I'll tell you a little bit about some of our work in this area. And unique to this discussion, many of these efforts were motivated directly by quantitative imaging and our desire to image glutamine metabolism. There are, of course, other strategies. I'll focus mostly on PET, but I'm very excited about opportunities with technologies like hyperpolarized MRI, where we might be able to have some complementary readouts given, for example, a PET or MR combined experiment. So let's look at some of the targets. We'll talk a little bit about some of these in some detail today. Two primary targets that our lab has focused on include SLC1A5, that's the gene symbol for ASCT2. This is the primary glutamine transporter that cancer cells use to import glutamine into the cell. Some other notable transporters that are involved in that process or related processes include LAT1, of course, which is thought to be co-expressed with ASCT2 in many tumors, as well as XCT, and we'll talk about XC or XCT some as well. This is actually a glutamate exchanger that will exchange glutamate for cysteine biochemically. Another target that's been the focus of pharma to a fair extent has been glutaminase. This is a mitochondrial enzyme that's involved in TCA conversion of glutamine to glutamate. There's been small molecules have been in clinical trials. There's been some companies. There's been some recent changes in company strategies around this recently around this target. But this is a compound that we've engaged in clinical trials with and developed imaging strategies around identifying patients who might be good candidates for glutaminase therapy. And I'll talk just a little bit about an ongoing clinical trial. So I thought I would talk a little bit about some of our work in the area of glutamine transport inhibition. So through a medicinal chemistry program that I won't go into today, but is published, we developed, identified V9302 and a few classes thereof of glutamine transport inhibitors. These compounds we identified originally as PET tracers because we were motivated to develop a specific PET tracer for ASCT2 because this had been identified by a collaborator at Vanderbilt as a marker of adenocarcinoma of lung and especially within the context of indeterminate pulmonary nodules. We then took that chemistry forward as we identified these as somewhat potent inhibitors, and ultimately through medicinal chemistry realized 9302 and members of that class thereof. What this compound does is it actually antagonizes the zwitterion recognition domain of ASCT2, and we can see that compound in antagonizing the active site as shown. We've gone on to further development of this series, but we pulled this compound out to publish to show proof of principle. And we found this to be both a potent inhibitor of glutamine accumulation and also an effective at limiting the tumor growth in multiple animal models. And so I'll talk a little bit about some of the features of this compound. And so initially, as we further evaluated the effects of this compound, we were first able to show that it was a bona fide inhibitor of ASCT2. This is a DARTS proteolytic degradation assay that shows that in a concentration-dependent manner, we can thermodynamically stabilize ASCT2 specifically in the presence of proteolysis. And this is a known assay for target engagement. We can also inhibit selectively glutamine within the context of other amino acid transporters, and that's what's meant to be seen at the bottom. And these were at relevant dose levels. We profiled the efficacy of the compound and its ability to inhibit glutamine transport across a panel of clinically relevant human cell lines. I've pulled out here those from lung, breast, and colorectal as representative of sort of the big three solid tumors. But we've done a number of others that were reported. And as you can see, we have selective potency across, actually in this panel, sort of a colon-centric panel. We could inhibit RAS and RAF mutants. Not all cells are sensitive, as might be expected, but many are. We could also inhibit patient-derived organoids, as we developed it at Vanderbilt. Some of the consequences of blockade of ASCT2 inhibition were guided by earlier efforts to knock out or to downregulate ASCT2 had been shown previously. And the primary effects are on mTOR signaling, some abrogation of MAP kinase signaling, an inhibition of glutathione, which leads to elevated reactive oxygen species, and autophagy were the principal responses that we saw across a large panel of cell lines. And so getting back to PET, if this compound in vivo were to be useful, or at least be on mechanism, we should be able to block the accumulation of glutamine in the tumor. And so we turned to the 4-fluoro analog of glutamine, and we could show in vivo xenograft models that we could, within a therapeutic window dose, we could fully block glutamine accumulation in the tumor to background levels. And we could use PET to do that. And that was actually an important measurement because this helped us to use our dose finding strategies, as well as evaluate occupancy, as well, and durability of inhibiting the target. So with that in mind, we went forward with a dose that was within the therapeutic window, and we went on to treat a number of models that Mike reported in this paper, and we could either prevent tumor growth, or we could reduce tumor volumes with a dose. And I believe this was 25 milligrams per kilogram twice a day, as shown here. Again, in vivo, we saw similar on mechanism effects, inhibition of the mTOR pathway, as well as upregulation of apoptosis through caspase 3 cleavage, as well as reduced Ki67. So, so this was an example. And we could also combine 9302 with other inhibitors. This is showing now a combination between 9302 and the EGFR targeted antibody panitumumab, and this led to some hypotheses related to our project in the GI spore, which I'll talk a bit more later, but within the context of colorectal cancer, combining an inhibitor of glutamine metabolism and EGFR. So going forward, what our plans are at MD Anderson, we're very excited, is to continue to sort of benchmark the 9302 but we have additional leads from this 9000 series. We've also developed a couple of other chemically distinct series that are on mechanism, and we'd really like to advance a molecule to candidacy. But within the context of an imaging project, I always like to point out that our identification of this particular series of interesting molecules, I don't think we would have found these had we not been looking for the PET tracers first, because we wanted to actually image that target. And so that was really motivated, our efforts were motivated by imaging first and foremost. So I'd like to talk a little bit about moving this forward now. We've been fortunate to be involved in Vanderbilt's gastrointestinal spore for a number of years. And when I relocated to MD Anderson, in very collaborative efforts, we've continued Project 2 as a multi-institutional project, and we've moved that forward across both those centers. And so I'd like to talk a little bit about that project. And again, I alluded a moment ago to the fact that our efforts to identify ways to overcome resistance to EGFR therapy in colorectal cancer, particularly in the setting of wild type KRAS, but recalcitrant to EGFR inhibitors. And so we had identified in the laboratory that there's significant interplay between glutamine metabolism and EGFR. We weren't the first to observe that. It's been known for some time. And in fact, glutaminase activity requires an active MAP kinase, and that's been known for many years. And so our thought in the spore was to capitalize upon blocking the EGFR with the antibody and blocking glutamine metabolism in the clinic with CB839, which is Calithera's small molecule glutaminase inhibitor. And the thought is that combined inhibition might resensitize or overcome resistance to EGFR inhibitors in those patients who did not respond initially to the antibody. And we also, of course, in the laboratory have continued to test 9302 as an alternative approach in that we're blocking glutamine metabolism well upstream of glutaminase at the level of glutamine transport. So there's a lot of research within this project. And so there's sort of the therapeutic side and the trial, but also understanding how to predict patients who might respond to these types of therapy. And so I'll mention just a little bit about that. So one trial that we brought with us to MD Anderson that we've been fortunate to onboard here recently and is just opening up is a study that builds upon the hypothesis that those colorectal cancers that are resistant to EGFR therapies, but again in the wild type RAS setting, will be glutamine avid. And so with Scott Kopetz and Christine Parsegian, we've opened a new trial asking just that question. Are glutamine avid tumors resistant to EGFR therapy? We were very fortunate that Scott and Christine have a wonderful trial that's an EGFR re-challenge with panitumumab in patients that are precisely in the clinical setting that we had originally proposed in Project 2, again, wild type RAS, but previously recalcitrant to EGFR inhibitors. And so we look forward to imaging these patients' baseline with PET. We'll use carbon-11 glutamine and another tracer, FSPG, that I'll describe in a moment, baseline, and then following them over time for survival and tumor volume change. So mechanistically, we developed the preclinical rationale for this combination, again, in the resistant setting, and this work was recently published by Alison Cohen in our group, but we could show that the combination of an EGFR inhibitor and the glutaminase inhibitor, CB839, could overcome acquired or intrinsic resistance to EGFR inhibition in the preclinical setting. Again, like we saw with 9302, our molecular determinants of response appeared to be reduced proliferation mTOR signaling, reduced mTOR signaling, and apoptosis. And so we're challenging in Project 2 this hypothesis using a number of preclinical models. We've assembled a very nice cadre and brought these with us from Vanderbilt to wild-type RAS colorectal cancer PDX models, again, across genotypes that recapitulate the clinical characteristics of the patients that we see in this setting. And we've also been fortunate to partner with Scott, who has many more PDXs here at MD Anderson. So we've been able to enrich this population quite nicely. We're treating these, of course, with our therapeutic strategies, cetuximab or panitumumab, and we have resistant and responsive models. And again, to develop the imaging footprint or fingerprint of response, we're using preclinical PET with both glutamine and FSPG in the preclinical setting, combined with a gene signature approach to really understand the molecular basis of the imaging phenotype and response. I'll jump ahead of that and tell you more about the clinical trial. So this trial is GI 1703, which is a phase 1/2 study to evaluate the safety, efficacy, and response of novel imaging biomarkers. Again, CB839 in the combination with panitumumab and irinotecan in metastatic and refractory RAS wild-type colorectal cancer. We've completed phase 1, and we're well into phase 2. The phase 2 cohort is 29 patients, and we've done now about half of those, 13 in our last count. We've seen safety, and we've actually had a couple of partial responses, and I'll just bypass these statistics in the interest of time. The rationale behind imaging in this approach is that we really clinically, I still don't think we know how to pair patients with glutaminase inhibitors. And so our thought process here was to take a multi-pronged approach. We'd like to ask a very simple question: do patients who respond to glutaminase inhibitors have tumors that are glutamine avid? We used a glutamine PET tracer for that. And then we'd also like to look at sort of a PD marker downstream of glutaminase activity to look at, did we hit the target or could we infer that? And we used a tracer called FSPG. So, at Vanderbilt and really anywhere, these patients are very precious and their time is very precious. And so we have tried to pair the radioisotopes appropriately such that we can capture a lot of information in one trip to the PET center. And so we onboarded carbon-11 glutamine in this approach, followed by the FSPG and F18-based tracer, so we could get both tracers because of the short half-life of carbon-11 in a single trip to the PET center. So again, carbon-11 glutamine is used for glutamine avidity. FSPG, again, a little bit more about that tracer. It's a substrate of the XCT, which this antiporter will exchange glutamate, intracellular glutamate, largely for L-cysteine. And so in that way, it's a measure in the case of this colorectal cancer setting of sort of this intracellular glutamate pool that's downstream of glutaminase, and may also give us some indications about reactive oxygen species that we can infer given glutamate and cysteine's role in glutathione production. So again, the idea is to look sort of upstream and downstream of glutaminase, and maybe we can get an idea. We did validate in preclinical models that by blocking glutaminase, we could infer and quantify distinct PET imaging phenotypes. Notably, in the pharmacodynamic regime, blockade of glutaminase results in an intracellular increase of carbon-11 glutamine in the tumor, and a decrease of FSPG. And again, that's in the pharmacodynamic setting, so we could validate that phenotype. Clinically, the time frame of these, we have to be careful with how we interpret the data, because in our trial, as it turns out, because of a trial-related protocol, we withheld medication of the day of imaging. But if we could validate this preclinically quite easily. So here's our schema again. So we're collecting pre and post, both glutamine and FSPG PET. We're getting both PET tracers in a single visit to the PET center. We get pre-treatment and cycle 2, day 1, post-treatment PET. Those patients then go on to standard of care imaging to evaluate tumor volumes. And we're collecting a number of correlates. We have a biopsy for tissue-based correlates, as well as some exosome analysis overall as well. We onboarded for the first time carbon-11 glutamine as part of this trial. And so this work has been submitted to Journal of Nuclear Medicine recently. This is one of the first whole body PET scans of carbon-11 glutamine, and gives you an idea of the biodistribution of glutamine in a human being. We have very low accumulation or uptake in the thorax, some mild uptake in the liver that you can see here, physiological uptake. We have some pancreatic uptake as well. So one should be aware of this when looking for tumors. This patient in particular does have a lesion in the left shoulder. You can see there. I don't know if my pointer is working, but that's where it is right there. So you can see that. So this has now been submitted. We have a number of imaging features that could be visualized in the trial as well. These are some lung metastases shown on the left hand. Again, we have some physiological liver uptake. We did observe some CNS lesions as well that actually were quite easily visualized, as the one in the lower right. The bio-D of glutamine, as you might expect, is primarily urinary excreted. We saw, as you can see, significant pancreas uptake, so we probably won't be imaging primary pancreatic cancer with this tracer, but this has also been seen before in the mice. Some liver accumulation as well, but actually rapid clearance, and the pharmacokinetics of the tracer are such that we plateau and reach steady state very, very quickly. So glutamine actually for an imaging study is actually very easy to use. So I'll pull out a couple of features of 4 patients that that have completed both pre and post imaging as a way to highlight the potential utility of this approach. You know, a lot of the subjects enrolled in these trials have synchronous lesions, just like patient 1 here is shown. And we're seeing very mixed responses by both the tumor volume changes, as one might expect, but also in their PET imaging characteristics as well. So patient number 1 came off trial as having progressive disease, but the 4 lesions actually are quite interesting if we look. And so if we look at the lesion in pink, this was a lesion in the right hepatic lobe that really was quite stable in its response to therapy. And if you look at the imaging characteristics with both glutamine and FSPG, we found that both were rather significantly reduced. Unlike the other 3 lesions where one tracer might be reduced, usually FSPG, indicating that we hit the target, but the glutamine was not substantially changed, indicating that we had not changed the glutamine phenotype. And in fact, that adrenal mass went up by about 30%. In fact, that adrenal mass is one that we're concerned about in some regards, because both the glutamine went up, but FSPG was really not changed. And so we're not even sure that we hit the target in that particular mass from PET. Here's a patient that also had progressive disease. Both lesions that we measured and followed were up about 25%, 20 to 25%. Glutamine was elevated in both lesions after therapy, and FSPG was decreased, indicating that we probably hit the target here, but we didn't really impact the glutaminolytic features from the glutamine. Here's a subject that was progressive disease, but notice their imaging characteristics. Both of the lesions that we evaluated would have been called stable disease. Excuse me. And their imaging characteristics from both carbon-11 and FSPG were significantly down. And in fact, the progressive lesion that this patient came off study for was not either of these two lesions. These lesions were actually quite well controlled. And then here's a partial responder where all lesions were either modestly decreased or significantly decreased, as in the case of the hepatic metastasis that largely almost completely went away. All of the imaging characteristics were very promising with both carbon-11 and FSPG. So, we've only done a few patients now in the study, but we're starting to see, that both tracers probably are useful in this setting to look at both sides of the pathway, but we need a lot more data to tell, really. And so we're hopeful that after COVID and the situation is we get a bit back more to normal, we'll begin to finish out our accrual. And so again, we found that imaging is well tolerated in this setting. So moving on, I'd like to talk a little bit about a diagnostic project that we've just started in hepatocellular carcinoma. And so this project is based in the setting of differential diagnosis and staging of HCC, which of course is a very common and the frequency is increasing malignancy of the liver. In the current setting, certain HCCs are pretty easy to call using standard of care MRI techniques, but those lesions that are in the 1 to 2 centimeter range can still be quite challenging. And notably, PET is not used in this setting largely because FDG is not sensitive to low-grade disease. There's a number of dual tracer approaches that have been used in Asia, but these are hard to implement. Carbon-11 acetate is used. And so we asked a simple question, could we use a different tracer as a single tracer and both improve diagnosis and staging in this setting? And so again, we're in the setting of FSPG. This is again a glutamate analog. It's a substrate of the XCT transporter. And there was some pilot data coming out of Korea that suggested that XCT would be a good target in the setting. We partnered with a company, Piramal, that later became Life Molecular, and we're one of a small group of institutions that have carried this forward in the clinical setting. But again, the idea, so we conducted some preliminary analysis of XCT and in HCCs in a large clinical bank assembled by K. Washington at Vanderbilt, and found that XCT was elevated in many of the HCCs across grades. So it looked promising. We also completed a pilot clinical trial of the FSPG in the setting. This is published now, but this particular individual was very characteristic of our motivation to use PET. And so this individual came to Vanderbilt with an outside MR read indicating 2 lesions in the liver. The top 2 red arrows are the lesions that were called on the MR report. We scanned the patient with the FSPG and we identified the small lesion that had been not observed in the initial report. And notably, in sort of the Milan criteria and LIRAD setting, if this had been a 4th lesion, this patient would not have been eligible for a liver transplant, which they got. And we found that small lesion was indeed a high grade but small HCC. And so again, we think this could be useful in the setting. We've done comparative studies with acetate, which is very thoughtfully and well used in Asia, but it marks the low-grade disease. And so the strategy that's been really pioneered by CL Ho in Asia has been to pair acetate with FDG to sort of try to capture a large swath of this disease. Acetate is again a 20-minute half-life, and it's hard to deploy, and the images can be hard to read. And you can see this is the same patient scanned with both tracers, and you can kind of make out one of the lesions with acetate, but the other two were not able to be visualized. And so again, trying to replace this with a single tracer, and as you might infer, the bio-D of FSPG is very, very nice for liver imaging, very low uptake in the healthy liver, even in the setting of cirrhosis. You might also see renal excretion here, as well as pancreas uptake. But we're very encouraged by the signal to noise, as well as the uptake in background tissues with this tracer for this particular trial. These were some of the metrics that we obtained in the previous trial. We scanned a total of 11 patients that we reported in that setting, and that motivated our new grant. So we've opened now at MD Anderson, supported by a new R01 grant, what is actually the largest trial of FSPG in a single cohort so far. We have 3 cohorts that are our primary cohorts. We have a resection cohort and a liver transplant cohort. We're very excited to have a liver spore at MD Anderson, and Ahmad Qassab will be assisting us with this trial. We also have a benign cohort for sense and spec. And then we have a bucket cohort that's not shown here of other tumors of the liver that we can get some additional information about non-HCC but malignant disease in the setting if we can accrue those from this particular trial. But we're very delighted. We'll be looking at repeatability in the transplant cohort as well, and a number of biological correlates in this setting. So our primary objectives and metrics are to evaluate PET with standard of care MRI. We'll be comparing PET to 2 tracers. So we'll be looking at FSPG compared to acetate, as well as FDG, which is not standard of care, but is sort of the clinical oncology standard. And so it's been included. It's what's done in combination with acetate elsewhere as well. So we'll have PET to PET to PET comparisons, RAD to PATH. We'll also be conducting some metabolomics part of the study as well to understand some of the basis behind the FSPG accumulation. Some of the things that we'll also be excited to partner with some of our colleagues from MD Anderson to look at include immune cell contributions to FSPG uptake. Effects of immunotherapy, I think, will be very interesting, especially as they pertain to redox, as well as the metabolic state versus the effectiveness of therapies or bridge therapies. And in fact, there's a large clinical population here where liver stiffness is being looked at, and how that might affect the metabolic state. I think will be very interesting as well. And then maybe one more short vignette would be, some work to identify opportunities in pancreas. And so this is a clinical setting that we've been interested in for some time now. The setting is the indeterminate lesions of the pancreas, and primarily the cystic lesions. And so as we know, in pancreatic cancer, diagnosis is usually very late. Imaging is very challenging and hard to deploy. But a potential low-lying fruit opportunity resides in these cystic lesions that are largely identified through routine imaging of something else. And so one of the conundrums that comes up with these is, so we have a lesion, but the anatomical features of that lesion by maybe CT or MR do not lend themselves very well to the future behavior of this lesion or how aggressive it is. And so the surgeon is left with this question, should I operate on these patients or not? And we've been fortunate to have collaborations with a number of outstanding pancreatic surgeons, Napun Merchant over the years, and Cameron Idries at Vanderbilt, and we look forward to more collaborations in this setting. But if we had a molecular imaging strategy here that could tell us about tumor future lesion behavior or tumor behavior that could be very helpful potentially in the setting. So Allison Cohen from our group has recently reported a biomarker we published in Clinical Cancer Research. I'll tell you a little bit about that study. So a number of years ago, supported by the Lustgarten Foundation, we identified translocator protein, which is associated with cholesterol metabolism, as a predictive biomarker in the setting of pre-malignant high-risk pancreatic cancer precursor lesions and pancreatic cancer. And this shows the correlation between TSPO and multiple grades of pancreatic cancer. There is certainly some TSPO positivity in chronic pancreatitis as well, but compare that with the normal tissue and that of tumors, and it's very modest. And so this, for tumor, this looks to be good. We looked at pannins as well, but I won't show you that data because pannins are small and this really would be a challenge to image those, as you know. But again, the IPMNs or the cystic lesions are the focus here because these are something that a surgeon could potentially might be actionable. So we found that IPMNs were elevated significantly, and the TSPO score with histology was correlated nicely with the grade. And so these were from tumor microarrays that were a combination of tissues collected by Chanwan Shi when she was at Vanderbilt, as well as Mike Goggins from Hopkins. We because of the limitations of tissue and TMAs, we tested this hypothesis in the mouse. So we developed or derived pancreatic models, the genetically engineered models. This is Hal Moses mouse on the right, TGF-beta type 2 heterozygous mouse, and also we have the homozygous as well. We could see that TSPO was elevated in high-grade pannins and ductal adenocarcinoma, but in low-grade disease, such as in the KRAS-only mouse, we found very low TSPO expression that was more analogous to normal tissue. Our favorite mouse in this setting was one that was described by Nabil Bardisi, which is a SMAD4 flox mouse. This mouse recapitulates the IPMNs very nicely, and these mice progress from a cystic lesion phenotype to adenocarcinoma in about 9 to 16 weeks. 16 weeks, most of them, the mice are dead at that point. We found that TSPO was elevated in very young mice in this setting. Very low-grade disease, but disease that was destined to become pancreatic cancer, as you can see in this 4-week setting as well as the 9-week setting. Through this grant, as well as some NCI funding, we had developed a series of translocator protein ligand PET tracers that had very good pharmacology for imaging tumors, particularly in the abdomen. Dawei Tong in my lab and many others have developed over the years some of this tracer technology. And this is using PET now in the SMAD4 deleted mouse. You can see that PET localized with MRI using a compound we call 1008 illuminated the tumor very nicely in this setting. And we could see in a cadre of mice elevated accumulation of these tumors in these mice. And so the idea is if a surgeon like my friend Kamran Idris had a patient that didn't know what the future behavior of this lesion were to be, could he then layer in a PET scan with this anatomical image to further galvanize his decision making for this patient cohort? But could he also, who being a surgeon, go one step further to utilize TSPO molecular imaging at surgery? And so a wonderful student of mine, June Lee, developed a very high affinity TSBO ligand that was based on the 1008 pharmacophore. We call it 1520. June made a large series of compounds to identify one where the ligand would engage the TSBO binding pocket, yet had the requisite linker distal to the binding, distal to the pharmacophore, so that binding could be very high in this setting. And so June developed this compound. It has about a 5 nanomolar binding affinity to TSBO. And then he went on to show that this compound could be used to profile and identify the tumors at a very early stage. It could be blocked. The signal is nicely blocked and attenuated, and could be was localized in very early disease as well as very late disease. Along the same lines, we started to do some image-guided surgery shortly before I relocated from Vanderbilt to MD Anderson, and using a camera from Curadel, we were able to follow the uptake of the tracer using image-guided surgery. And I have these images shown here looking at the healthy pancreas, as well as some tumors on the bottom, and the healthy pancreas is on the top. You do see that this tracer has some clearance and uptake in the liver. So we won't be looking for liver mets, but to look actually at the primary tumor in the pancreas. The BioD at the current stage looks pretty good. I think I have well, here's some more mice that were just part of that group. So the mice on the left do not have tumors, and the mice on the right have tumors. And you can see we could localize that in vivo as well as ex vivo using this is an Odyssey scanner. And so these are mice, these are videos. I hope they play. So this is a control mouse. So again, we can do this in real time. We were very fortunate to partner with some wonderful mouse surgeons at Vanderbilt that were part of Ming-Feng Bai's lab. And so this is a control mouse. Again, we can already see the liver is fluorescent, but we're not going to worry about that. And we're going to open up, we can see some intestines shown here, and the pancreas is coming. So the white tissue that you see there is going to pull that out. That's going to be the healthy pancreas. We have very little fluorescence. So you can see the spleen as well has very little fluorescence shown there. And then I'll show you the tumor. Now this tumor is unfortunately a fairly large one. I'd rather show you a small one, but we're just getting this study back up and running here at MD Anderson. This is pretty recent data relative to the time frame of our move. So again, live mouse. And we can see here comes the tumor. So the tumor is nicely green fluorescent. So we got a long ways to go with this particular project and tracer, and certainly near infrared imaging is a higher bar than is PET. But our plans are ambitious. We'd like to start simply though by really validating the PET. And so we've started the process of opening a trial here at MD Anderson. Anirban Mitra is one of our key colleagues in this trial, and we're going to look at TSBO PET in the setting of cystic lesions. We have some tracers that we've developed in-house, the 1008 and another one's 8310 that have somewhat higher affinity, but we're going to start with carbon-11 PBR28 because it's been in the clinic before, and we can sort of do our sanity check clinical trial early with that one. So that was the idea. But we'd like to continue the surgery as well. And we recognize that as a higher bar, but we're very optimistic to advance this paradigm forward. And so with that, I hope I've left a little bit of time for questions. I have just so many people to thank, many friends and colleagues who are still at Vanderbilt. I was Vanderbilt in total for almost 20 years, including finishing graduate school there and then through postdoc, and many good friends in the Cancer Center and the Imaging Institute, but also making a lot of new friends and colleagues here at MD Anderson as well. we're thankful to all of our funding agencies and support. And I thank you for your attention today and taking part in this in the part of the seminar.
Thank you, Dr. Manning. Great talk. I want to invite the audience to either enter questions in the chat or also to just unmute yourself and ask any questions that you may have.
Hey, Charles. Hopefully you can hear me. This is Jack. So as you may remember from our time together at Vanderbilt, I was really interested in the pancreas, not looking specifically at pancreatic cancer, but what's happening to the pancreas and diabetes. And so I was curious about your glutamine tracer, where you see that really high uptake in the pancreas. Have you looked into that at all and know whether is it exocrine uptake that's part of protein synthesis, or is any of it part of endocrine uptake? I think there's some literature out there that glutamine might actually be a possible treatment to reduce or to improve insulin secretion. So I was curious if maybe you've done some autoradiography on the pancreas and been able to kind of parse that out a little bit.
We have not chased that directly, Jack. Thank you for the question. Thank you for coming today. Our current thinking is that the pancreas is hungry for metabolites, and this is a feature of that. But we have not chased that down to any certain extent, but would be interested in doing so. If there's something that we could partner with, I think that would be great.
Great, thanks, Anna. Maybe we're both in Texas. We can try to figure that out together.
Absolutely.
Hey, hi, Dr. Manning. This is Roby Petler. I also have a follow-up question. Just wondering, for the glutamine distribution, have you observed if the baselines change during aging?
Oh gosh, aging. That's a very good question. We have not done enough patients to say that, that would be very interesting to know. One of the limitations too of our studies and all these baseline measurements is unfortunately when our subjects are enrolled in these studies, they've kind of had a long road, and so there's the baseline characteristics sometimes can be quite different, than sort of a healthy person. So that's a great question. I'd love to know the answer. I can tell you preclinically, we have some experiments that we're getting started here to understand some features like that in mice. Aging is certainly one of interest. We're even thoughtful about things like light-dark cycles, chronobiology and diet. We've seen that diet does in fact impact glutamine in mice. Don't know how well that translates over to humans, but we know that there are characteristics of imaging protocols that can give us more or less glutamine accumulation in tumors and normal organs. Don't have the aging question exactly answered, but that is something of interest. So I appreciate the question.
Maybe this is Jack again, Charles. Related to that last question, I know early onset colorectal cancer is really of interest, and there's a thought that maybe it's molecularly distinct from kind of late onset disease. Have you looked at either patient imaging or PDX derived from early onset disease versus later onset disease, and do you see differences in glutamine uptake across those two?
That's a really good question. I'm going to write that one down. I don't know that our models, I don't know that I have that information about our models. But that's a really good question. And unfortunately, I don't know the answer. But you're right that there could certainly be metabolic characteristics that are different. Some literature in the sort of oncology glutamine space would suggest that glutamine is a feature, right, of sort of MYC, and RAS can be mixed in. I think that's tumor, specific, right? And so if we're talking about lung or colorectal or breast, those may, some of those features may not cross over from tumor to tumor. And certainly we find in colon, some of the interesting features colon being a disease of sort of acquired mutations over time, two colorectal cancers that seem to be very sensitive to our inhibitor are those of RAS or RAF, interestingly, genealogy or genotype. And of course, the wild type RAS are a focus group in our trial, and we're testing that. But the RAF group was one that emerged very early. And so I'm not sure that's helpful towards answering your question. That's more of a rambling answer, but your point is well taken, and we'll have to look into that.
Any other questions from the audience? Well, I'll go ahead and just ask one. I think the audience might be interested in hearing from you now that you're here with, several institutions as part of this audience. What kind of collaborations do you think you're going to be seeking as you get your lab set up? I know you mentioned working with different surgeons and, really kind of wanting to move into this, into this pancreatic cancer study and these precancer lesions. What else are you looking for as far as collaborators go?
Oh gosh, yes. So we're working in the area of sort of diagnosis. That would take us into the surgery space, pancreas, liver, colorectal. Treatment response takes us in many different directions. Very interested in how to predict tumors that respond to therapy. I'd like to develop, an underlying basis for what gives rise to a PET tracer phenotype. I don't think that we really know that, so we're developing gene signatures to sort of extend the utility of PET, perhaps to centers that don't have, for example, glutamine PET. We're developing new radiochemistry technologies. So if there are engineers and those that are interested, particularly in microfluidics, one of the frustrations with PET and radiochemistry in particular is the cost. So we're trying to develop new ways to prepare individual doses of tracers. We've developed some microfluidics work. I didn't have a chance to talk about that today, but Leon Bellin at Vanderbilt is our collaborating engineer, and we'd love to extend that to the Gulf Coast region. So those would be some areas. I'm very fortunate to have landed at MD Anderson in a time where theranostics is a huge interest. There's many groups working in that area, and so that would be certainly of interest as well.
Well, thank you again. We really, really appreciate you joining us and look forward to hearing more from you as we move forward. Like I mentioned, we're definitely going to have theranostics as part of our next imaging conference. So excited to hear how things are progressing. Hopefully you'll have your lab set up and all your folks hired and be moving and shaking. So we really appreciate it.
I hope so. And thank you so much for the opportunity. And I appreciate everyone for their attendance today.
All right. Thanks, everyone. Y'all have a great rest of your day.
Okay.
Bye-bye.
Bye-bye.